Hello again, everybody. My name is Emily Perry, and I was here last week for the Genomics Genome Browser session. I'm back again to talk about human genetic variation. So the learning objectives for today's session. At the end of this, you should be able to distinguish different types and consequences of genetic variation. You should be able to differentiate between major human genetic variation sequencing projects. You should be able to describe the types of analyses that people do with genetic variation. You should be able to read genetic variation data in commonly used formats, including VCF and HDVS, and you should be able to express your own data in these formats. You should be able to synthesize the considerations involved in variant prioritization from a variant calling study, and you should be able to carry out variant prioritization using data from the Ensemble VEP and related tools. So the first thing to mention is variant types. So the first classification we do is on scale. We have small scale variants, which affect sort of one or a few nucleotides of a gene. These include small insertions and deletions, as well as single nucleotide polymorphisms. And large scale variants, also known as structural variants, which include copy number variations, large scale deletions, duplications, insertions, and translocations. We categorise variants as well by how they affect the genes that they hit. So this sort of simple diagram of a gene shows us some of the various places that variants can fall, and so we categorise whether they fall within the coding region, whether they change the coding sequence or not, and how likely they are to have an effect. The standard terminology, I don't expect you to be able to read this, but the standard terminology used comes from sequence ontology. And so that this terminology is used throughout to describe these different consequences that variants can have on genes. And this is the terminology used by Ensemble. This is the table that Ensemble use. And there's also colour codes that Ensemble use throughout for these different types of consequences. A rule of thumb that you can use in Ensemble is the kind of danger colours like red and orange and yellow tend to be put on variants which are likely to have a significant effect on the protein function, whereas the sort of more safe colours like blue and green tend to be put on variants which probably don't really do so much. We also commonly use algorithms such as SIFT and polyphen to predict the effect of missense variants. So missense is where you change one amino acid for another. And SIFT and polyphen can predict how likely this is to actually change the protein. So this includes how well conserved the protein is. So if it's a highly conserved protein and there's always a particular base at a particular locus, we think changing it might have some major impact. If there's a chemical change in the amino acid, so if we switch out a positively charged amino acid for a hydrophobic one, for example, this may completely change your protein structure, whereas if you switch one positive for another positive, it's likely to stay roughly the same. Polyphen also takes into account 3D structure and known domains. An important thing to remember whenever you're working with algorithms like SIFT and polyphen is that these are predictions. They are not facts, they are not experimental evidence. They are just pointers towards, if I found something that looks like it's got a deleterious SIFT and polyphen effect, probably means I should do an experiment to confirm that. It doesn't mean that that is definitely the case. Indeed, you could switch out a positive for a positive, but it turns out that's actually found in the active site or something like that, and so really important. SIFT and polyphen both give you a score out of 1, and really annoyingly, I don't think the people who made SIFT and polyphen talked to each other at all because their scales are opposite, and I never remember which one is which. Fortunately, they always come with predictions as well, so these are just the scales that they use. So anything in SIFT that's less than 0.05, they count as deleterious. Anything above that is tolerated. Polyphen, the cutoff is between 1 and 0.908 for probably damaging. then they have possibly damaging and benign. And you'll never see the scores without the predictions with them as well. And polyphens sometimes change these boundaries. And if you're using a different version of polyphen, then the predictions that you'll get will match whatever was the chosen predictions at that time. Ensembl has variants from a number of sources, and there are loads of databases of variation data out there. dbSNP is the major one for human genetic variation. There is an equivalent database, EVA, which takes in data from other species. But there's also lots and lots of data which is tagged onto variants, such as phenotype data and other information about it, which you can find from places like OrphanNet, OMIM, the GWAS catalogue, for example. There is a lot of data about human population genetics, and here are some of the projects that are available in Ensembl, where you can get allele frequencies. So HapMap was a project with worldwide genotyping. That then got superseded by 1,000 genomes. So this is worldwide healthy whole genome sequencing, and I'll talk a bit about the population breakdown in a moment. But the important thing about 1,000 genomes is that all the individuals in there are healthy, so it represents a kind of normal sample. They also tried to sample from all around the world fairly equally, although there are some populations missing. Nomad, and it's kind of it used to be EXAC, and then Nomad absorbed EXAC. This is mixed disease and healthy whole genome and whole exome sequencing. And basically Nomad and EXAC pulled in studies from loads of different sources, any kind of publicly available data for which they could get counts of alleles at particular loci. They pulled those in and added them to the database. So this means they have vast numbers of variants and vast numbers of individuals for which they can calculate frequencies, but it is skewed to the most studied population. So by a long way, the majority of data in Nomad and EXAC is from European individuals. There's also data in Ensembl from UK 10K, which is UK-wide, and TopMed, which is disease whole genome sequencing. So if we look at 1,000 genomes as an example, 1,000 genomes took approximately 100 people from each of these labelled populations around the world. And each of the populations is a subpopulation which forms part of a superpopulation. So all of the purple ones here are the African subpopulations. And you'll see, as well as populations taken from different regions of Africa, they also have a group from the southwest of the United States and a Caribbean group as well. These are obviously different to the African groups because of things like admixture and sort of sampling effect when these were taken. So they won't be as expansive as the actual African groups. And it's worth noting that the Nomad African frequencies, a lot of those will probably be African Americans rather than Africans. If we look at the data from 1,000 genomes in a bit more detail, and we'll see how you can find individual genotype frequencies and things, but this is just showing a kind of population view. And each of these pie charts represents, in the grey, variants that were found in this population that were found in all of the other populations. The lighter grey are found in other populations, but and in some of the other continents, but not all of them. This light yellow, these are variants that were only found in this example in the African population. This light pink was only found in the Indian subcontinent population. And then the darker colour represents variants which were only found in this particular population. And one of the really interesting things you can see from this graph and this map is that the coloured sections on these graphs are much larger in the African populations and very, very small in, for example, the European and the mixed American populations. And it's believed that this is to do with sampling effect and founder effect when the migratory parties left Africa and moved into other parts of the world. We then ended up with much less genetic variation within our population. So studying population structure in African populations is actually very different to studying it in some of the other populations. If you just look at variants per genome as well, sites which have bases different to the reference, there are far more in the African population than there are in most of the other populations, which is really quite interesting. And you could argue this is not because the reference genome is not African. Actually, the majority of the reference genome comes from an African American individual. It's actually because these people, the African people, do have more genetic variation in their genomes, which is really interesting. Going back to the idea that this genome, that I mentioned last week, that the reference genome is made up of sequences of different individuals. We have these contigs. If you look at any particular genomic region and you find a variant in that genomic region, taking this example here where we've got this T, and we're saying that this lady here, who is a stock photograph, but we're pretending that she's somebody who the reference genome was made of, she has a T at this particular locus. And that is still the reference allele. So it could be that this T that she has at this locus is very low frequency, and another base at that locus is much higher frequently. And it could be that the alternative allele is also the one that's the ancestral one, because it's found in all primates. It could be that the allele she has causes some kind of disease susceptibility. But because that's the allele in the contig used, it is classified as the reference allele, and the G is classified as the alternate allele. This is the standard convention that is used. The reference is always whatever happens to be in the genome. And we quote those alleles as reference/alternative. There's another point to make about allele strand. Variants always affect both alleles. A T/G on the forward strand is equivalent to an A/G on the reverse strand. And it may be, when you're looking at things like gene sequence, that the reverse strand alleles are the most relevant. The ensemble convention is to always store the forward strand alleles in the database, and I believe that this is replicated across by EXAC and Nomad and other sources. However, dbSNP will do either, but they will label them. The big danger is when you start looking in the literature, and in the literature they will often give you alleles, but they won't tell you which strand they're on. Sometimes you can work it out by what gene they're talking about and actually just looking at what's there. And that's fine for a variant like this, which is a TG or an AC. If you're looking at a GC or an AT, it can be very confusing and misleading as to which one might be the relevant allele, the disease-causing allele that they're talking about in this paper, if it's not clear, if they haven't told you which strand they're on. So do look out for things like that. It can happen. So we're going to have a look at a gene, and we're going to look at MCM6. We're going to find variants in the gene and in the region it's found on, and then we're going to look at an individual variant, RS4988235, and we're going to find out more information about it. And after that, there's a task for you to complete.